Hello, and thanks for joining us today for this repertoire webinar. My name is Matthias, and I will be your moderator for today. In this webinar, Dr. Matthew Johnson will discuss clinical trial results from the University of Minnesota demonstrating complete response to TIL therapy in a patient with metastatic colorectal cancer. Without further ado, I would like to pass the webinar to Dr. Matthew Johnson.
Hi, everyone. As was said, my name is Matthew Johnson. I am a scientist in the Brandon Moriarty Bo Weber lab at the University of Minnesota here in the Department of Pediatrics. And today I'm going to be talking to you about one of the patients from our recent clinical trial examining the use of CISH-knockout tumor-infiltrating lymphocytes for the treatment of advanced metastatic colorectal cancer. So this is mostly based on and presented in our work in our recent publication in Lancet Oncology, where we basically present the full results of the trial. And so here's just a general outline that we've used to kind of guide people through the trial. And we'll go through each of this steps in more depth, but in general, we resect the tumor. We will grow out TIL from the tumor, do a complex neoantigen screening. So we select out aliquots of the TIL that are actual, or fragments as we call them, of the TIL that are actually tumor reactive, edit them to knock out CISH, expand them, and then infuse the patient. But before going further, just to explain what the gene CISH is, the CISH gene is a member of the suppressor of cytokine signaling family, things like SOX1, SOX2. CISH is a member of that. And it has two well-characterized roles. One, it's a negative regulator of cytokine signaling for specific cytokines. It's involved in inhibiting JAK-STAT. And perhaps more relevant for this particular study, it is considered an intercellular checkpoint for T cells. And that's actually what the graphic over here shows. It functions through, so normal CISH function. We will have PCL gamma bind CISH, and we will get degradation of PLC gamma, and that reduces your TCR signaling. When we go to a CISH knockout, that CISH is not there to degrade the PCL gamma or PLC gamma, and we get enhanced signaling. And the diagram here in below shows the kind of the functional impact, the real impact of this. And what it does is, in effect, lower the activation threshold of these particular TIL. So less signal or can actually activate this the TIL much easier. And so to go through our trial design kind of step by step, step one is a surgical resection of the tumor. This would most often be from a liver or a lung section, where we would take this TIL and then chop it into 2 millimeter cubed bits and grow that in a G-Rex flask with expansion media and very high dose IL-2. From that, we would have a clinical aliquot, most of that, and then a research use only aliquot that we would then take to go to the whole exome sequencing. And just to highlight what's here, or what happened here, you can see here are each individual patient in the trial and the number of fragments that were deemed acceptable for downstream applications. So they're in kind of the salmon color here, and the amount that did not grow out high enough. As you can see, some donors fragments grew nearly 100% over that. Some donors, we actually had very few fragments that could actually proceed onto CISH or downstream applications. It really varied greatly between donors. And then you can see just the number of viable cells in each fragment. Oftentimes when we exceeded the 15 million cell cutoff, we greatly exceeded it. So now to go on to the neoantigen screening step, which is probably the most complex to explain, so I'll go a little into depth here. But we would do whole exome sequencing and RNA sequencing of the tumor, and then exome sequencing of a normal tissue. In this case, it was usually peripheral blood cells. and we would compare the exome sequencing to identify candidate neoantigens. So just individual SNPs that would result in an amino acid change. From that, we would synthesize the neoantigen peptide. This would be 25 mers with the number 13 position being the SNP, the changed amino acid that is present in the tumor tissue, but not present in the host tissue or the normal host tissue. This was because the largest you can fit on like a MHC class 2 peptide is generally about a 13 mer. So by using these 25 mers with the mutated peptide, mutated amino acid in the middle, we would have coverage of basically every possible peptide that could be made with that peptide or with that SNP. We then would generate using the patient's own cells, either an LCL line, so that's an immortalized B cell line, or monocyte-derived dendritic cells. Both of these can function as professional antigen presenting cells. And these antigen presenting cells can be used to then present antigen, our neoantigen peptides, into our TIL cultures, each individual TIL culture. And we can use ELISpot and flow cytometry to judge which cultures are reacting to the neoantigen and which ones are not, or candidate neoantigens and which there's not. So when we went through this, we had a very great very great differences in the number of candidate neoantigens detected from each individual in the study. You can see all the way down from maybe a dozen up to over 200. Interestingly, the complete responder was this UMN22, but we'll get into more detail on that in a bit. And this is an example of one of our neoantigen screening results. And then in fact, this is from UMN22. So what we would do is run an ELISpot, and each ELISpot would be run in duplicate. So top row is replication 1, this is replication 2. And we could look at interferon gamma or TNF production in the ELISpot. We would then run in parallel flow cytometry to look at activation markers 41BB, CD69, and OX40. And we have it labeled here because we didn't want to load several hundred peptides into a single well. That often would limit our ability to detect lower level responses. They would get shielded just because there's so many peptides. We actually broke the peptides into usually about 10 different pools so that to reduce the number of peptides we're presenting in each individual well. So you can see for this particular donor, we were able to get responses by ELISpot to pool 6, 7, 8, and 9 very easily, along with some signals in 10 and 5. So that's actually quite a bit of reactivity that we see here, mostly in pool about 8. And so what we would do is we'd find out of all the fragments we would make, those 2 millimeter cubed sections, we would select the ones that had the neoantigen reactivity, usually 1 to 5. In the case of UMN22, that was 2 fragments that we saw strong reactivity like that. And we would CISH edit them using CRISPR-Cas9 editing, and then expand them in a process called a rapid expansion protocol, which involves co-culturing the TIL with feeder cells and a bit of anti-CD3 antibody. Expand those for a couple weeks, and then infuse that into the patient. In general, we were able to achieve very good CISH knockout in almost all of our patients where we attempted this. You can see here, it's 70 to about 90% as measured by tide, and a little bit higher measured by Western blot. We do see a difference in the 2 methods, which we wouldn't think see as much of a difference, but this is consistent both with the trial samples and the samples we tested before the trial. It's just a bit of a quirk of the 2 assays. And the infusion dose would actually vary quite a bit between different patients. Here are the patients we were able to successfully infuse with the TIL therapy. Notably, the first 5 here, we were limited by FDA requirements to the maximum dose we could administer at the time. The latter 7 patients, we could dose whatever level we felt appropriate. That was always the highest dose that we possibly could. So you can see quite a bit of difference in the amount of TIL we're able to outgrow from individual patients. And again, notably, UMN22 here is actually a rather small outgrowth, quite poor compared to what we were able to do with a lot of other samples. And this is probably the graph I'm most proud of, having gone through this process a bunch. This is our production timeline. And what you can see here listed in red is the optimal production timeline for each of the steps with the actual production timeline. And as you can imagine, a trial of this size and this manufacturing complexity requires a lot of different organizations and moving parts and people. And so we have here time to biopsy to growing TIL, the optimal being 42 days, the actual being 42 days. We were able to do that successfully every time. When we ended the TIL growth to starting neoantigen reactivity screening, the optimal would have been about 10 days due to manufacturing issues and production availability for a manufacturing suite at our GMP facility. That extended out to about 20 days. Our GMP manufacturing of the actual TIL product, ideal would be 21 days, took 23 days. Lot release testing, ideal would be 14 and took 18 days. And then from this point, it was really up to the clinician when they felt the patient, it would be appropriate to infuse the patient. We wrote 14 days as optimal, managed to do it in 18 days. So kind of given the real life limitations of the manufacturing process, I feel we've done a very good job of actually doing this as fast as this particular process would allow. So now on the actual patient results from the trial. From this trial, we enrolled 22. One was ineligible for the study. All of that, 22 were consented and enrolled. One then withdrew before TIL collection. We collected TIL from 20 of these patients. 9 of them withdrew before we could infuse them with TIL. There are a variety of reasons for this, but most of this was due to disease progression, making it no longer possible to treat them at that point. Out of the 12 that were treated, we were able to analyze them for safety and for TIL efficacy and for the response to TIL. Out of this group, we had delayed progression of 1 month in about half of the donors, 6 of them, and delayed progression for 2 months for 4 additional donors. And then you can see here we have 1 donor, UMN22, to this day that is still not had any progression or survival, or is currently surviving. And in fact, we've actually had an update in the last couple of weeks that it is now approaching 3 years. He recently went through and is still not showing any signs of remission or I'm sorry, any signs of relapse or anything to that. So we're approaching the 3-year mark of what looks to be durable remission. And so the rest of the talk, we're going to focus in specifically on this patient with durable remission. This patient, you can see here, we have scans of them pre-treatment where I always have trouble seeing this, but I'm assured it's actually true. You can see tumor here at 6 months, it's completely gone. Similar here and here. And then this, I believe, here is completely gone again at the 6 months. As far as timeline, most of these the bulk of the tumor was completely gone by 1 month. The rest is, for what I understand, was gone, at least by this analysis, by 2 months. And we have not seen anything since. So that was very exciting. And we wanted to take a look then what specifically happened in the blood of this patient compared to our other patients. What we have here is proportion of the TC repertoire, or the T cells that fit into these categories. So it's CD4 positive or CD8 positive, PD1 positive or PD1 negative. And this is really quite interesting because we do have our infusion right here. And then at around 10 days, we have a spike in our CD8 positive, PD1 negative population, with a drop in our PD1 or CD4 positive, PD1 negative population. And then at 28 days, that flips. If we look at over here, the percentage of different TCRs in the peripheral blood, what you can see here is this is the patient's TCR repertoire at 7 days before we infused. And this is just looking at the top 10 TCRs present in the blood at the time. You can see the top 10 most common TCRs in the blood make up an extremely small frequency of the blood. When we go, and this is our TIL product here, so when we look immediately after the infusion, or 4 days, I should say, after the infusion, about 80% of the T cells in this patient's peripheral blood are of these 10 clones, and notably, these 10 clones generally match these, they're color matched. Our infusion product then drops off quite quickly to around day 10, where we get this second wave of TCRs that are generally not present in our infusion product at all. Or if they are, it's very, very low level. This is mostly, however, what we believe to be endogenous T cells or TCRs from the patient, not from our TIL product. Then at 28 days, we get another shift in the top 10 most common TCRs in the blood. And these emerge as high frequency, and the top 10 now make up about 50% of the peripheral blood. Similarly, these are not at all in our TIL. The most common ones are presumably endogenous TIL from the patient's immune system. And this percentage drops off down to about 12 months. Notably, this pattern and the timing of this, they actually do match pretty well at the switch at 10 days and then another switch at 28 days. And then we can also look at the percent of edited TIL we actually have in the blood. And that's actually one very exciting thing about this project is it's actually difficult for other TIL studies or previous TIL studies to know if a TCR comes from the TIL or if the TCR comes from the patient. You can measure the TCRs present in the TIL, but you can't measure every TCR present in the patient. So you cannot really rule out the fact that maybe you put in TILs with that TCR clonotype, but those TILs may not have outgrown that, may have just caused a patient T cell clone that matched to outgrow. With this study, because of our CISH knockout, we can actually measure the amount of cells in the peripheral blood that have our CISH knockout. And that data is shown right here. And so you can see this is the percentage of CISH knockout cells that fit in each category from that have that CISH allele. And so we have a spike at our TIL at around day 10. It's tough that we don't have a day 4 time point for this, unfortunately. And then seemingly spike a little bit in some populations at 28. And then up to 56, we can still measure CISH knockout cells in the patient's peripheral blood, which is quite interesting. Fortunately, with this rep-seq analysis, we were able to do quite a bit deeper analysis, and so that's what is here. So this is just the neoantigen screening results I showed before. As you can see, there are multiple peptide pools that look reactive, but in our mind, pool 8 here was probably the most reactive, and it's the one we had peptide and we had enough cells to test for. So what we could do is look at this. So we had 3 conditions. We had our TIL, and we broke them into 3 conditions. Those stemmed with peptide pool 8, those that were stemmed with no peptide, so just our control, and then a general OKT3 activation. By comparing these 3, we can identify TCR clones that are reactive to pool 8 specifically. And we can actually then take those TCR clonotypes we've identified here and measure their presence in the blood. So this data, B and C, is all coming from the leftover infusion product that we were able to test. And this data here is peripheral blood from the patient. And so by matching TCR clonotypes, we can see an increase at day 4, day 7, and then a pretty steep drop off, as has been noted before with the TIL. But we can track that some of these TCR clones, in fact, 21 of them, we can track out to about 12 months. And interestingly, they tend to cluster here, which is defined by 5 genes, expression of 5, upregulation of 5 genes. Specifically, we're just calling it the granzyme A cluster here. So when we actually take a look at those, these 4 graphs here are the TCR clones with the highest phenotype. We can actually look at CISH expression in those. And what you can see is CISH expression is generally lower in these cells than it is in the other pool 8 stimulated cells that we have. This is consistent with a CISH knockout if these cells did originate from the CISH knockout cell, our TIL group. We also have so this is, I guess, 17 other clones here. You can see that they also tend to be lower CISH than the rest of the pool 8 stimulated cells. And so to quickly summarize here, we've infused 22 patients, or we enrolled 22 patients, and we're able to infuse 19, 12 of those were dosed, 6 underwent delayed progression, and then we have the 1 patient with a durable remission out to, as of January, still durable remission, approaching 3 years, I believe, in March or April. And so to just go through the highlights of this, in general, these CISH-edited TIL were generally safe and well tolerated. We did see the expected side effects we were looking at, or we knew would occur. This is due to the preconditioning the patients needed to get prior to TIL therapy, and then also the high dose IL-2 that they received after therapy. Our CISH-edited TIL manufacturing was generally successful. We were able to manufacture TIL for every patient that we attempted it. 1 patient we had to go through 2 manufacturing runs, but we were successful. And then 1 of these patients had a complete and durable response following the TIL therapy. Actually was in the news quite a bit when we were first allowed to go public with it. That was very exciting. This patient demonstrated kind of 2 waves of nucleus TCR clonotypes that were not present pre-infusion or with the TIL infusion product, or at least not that we were able to detect. And this first wave started around day 10 and about day 28. So this may have been an example of epitope spreading within the tumor. And then that certain TIL groups that persisted beyond a year. They expressed this high granzyme A and then 4 other genes, I should say, and generally had very low CISH compared to what we saw with standard or more normal TIL. So our future directions for this project, we want to, for those 21 persistent clonotypes that we detected, we want to identify the cognate neoantigen peptide from them. This data could be valuable just as an idea of what characteristics these peptides or these genes may have. Similarly, we want to determine the characteristics of the TCRs and the cognate peptides for those 2 different waves. We have the TCR signaling pathway, we have the TCR sequence, but we don't know what neoantigen peptides they're actually reacting with. If they're reacting with neoantigen peptides, it's a possibility that these are just bystander TCRs that happen to expand a lot during the therapy. Luckily, we do know or have a very good idea of the neoantigen peptides present in the patient. So if we clone out these TCRs and these TCRs, we can actually just screen them against our peptide pools and get an answer as to what these neoantigen peptides may look like compared to the rest of the neoantigen peptides where we don't have a response mid-therapy. We do want to take a look at the potential roles for any of the genes that seem to be upregulated in that specific population that ends up persisting quite a long time. And then by far the most important and exciting to us is we want to develop a far faster manufacturing protocol that yields more potent TIL. I did mention that we had made product for 19 patients, but we're only able to dose 12 of them. I believe two or three of those patients were withdrew for different reasons, but many of them, most of them, became ineligible for the therapy due to disease progression. So although we were really quite effective in manufacturing our cells as fast as possible given our protocol, it still was not fast enough for many of the patients. So we're really focused on getting this quite a bit faster than that 100 days or so that it took us during this study. And then obviously we had delayed progression in many patients, but only one durable remission. So we need far more potent TIL as well. And so I guess this is the majority of our effort at this time. So going through this, I just want to thank people who were involved in the study. So at our group here, we've basically called our half of the lab the TIL lab for a while. The project was led by Brandon Moriarty and Bo Weber. And Lou was the clinician who ran the study, and then Tim Starr and Scott MacGyver were also heavily involved in um, basically every step of the study, developing the therapy, tech transferring it, filing an IND, and then actually running the actual trial, along with all these other groups that were helping us out quite a bit. As you imagine, it's a massive, massive process. Um, and with that, I guess I can take questions.
Thank you very much, Matthew, for such a great presentation. So, the first question that we have, Matthew, is considering that the tumor biopsy has blood and TIL infiltration, when you look for target mutated proteins to use as neoantigens, do you compare versus blood or versus healthy tissue? Since they could be mixed, what is the cutoff percentage you deem acceptable? And do you know if they are homozygous or heterozygous mutations? So I guess three questions in one.
Oh, sure. So the what we would do with the tumors is we would take the tumor and then send it to pathology, who would section the tumor into Z-stacked or multiple sections. We would then look for the portion of the tumor that had the highest tumor density based by a pathology. Because you are right, there is a mix of normal tissue, blood, or just healthy tissue more often than blood that is kind of contaminating that. And then the tumor, which we're most interested in. So they would basically try to use the sections, tracking the 5 sections below the section that we're interested in, the 5 above, to try to determine the tissue with the highest density. And that would give us the highest sensitivity for these mutations. Notably, it's not like when we sequenced this tissue, we would see 100% mutation rate. We would see varying levels, and we would have to have a cutoff for, well, is this high enough to be a true mutation that is present in the tumor? But yeah, very good question.
Perfect. Uh, Another question that we have is, did you see higher CISH-KO percentages in the UMN22 patient?
It was near the higher side. That was about 85 by measured by tide, and about 90 to 95 as measured by um, um, tide analysis. So that was near the high, but it was not the highest, and it was not unusually high, just a bit higher than average that we saw.
Perfect. Other questions that we have, what was the thought process to decide the peptide pool number to 10? And what is the number of peptides per group that you used?
It wasn't initially that we would always have 10 peptides or I mean, that we would only have 10 peptides. We tried a couple early donors where we would do more than 10, and the logistics of
Hmm.
managing that many spot plates and flow plates became very difficult. They're just it ends up being, I think one patient we had 28 plates, spot plates that were juggling simultaneously during the neoantigen screening. So as kind of a way of limiting that and avoiding potential errors, we eventually settled on we would do a max of 10 um, peptide or 10 peptide pools per donor. That started on donor, the third or fourth donor we went through. Um, the number of peptides per pool varied greatly for donors that had low neoantigen burden or candidate neoantigen burden. We would have one or even two peptides per pool. For UMN22, we had up to, I think, 25 to 30 per pool. Just that one had the by far highest candidate neoantigen burden. So it had necessitated the most peptides per pool.
Perfect. So, do you see any correlation of fragment appearance, pre-rep TIL growth, and the corresponding reactivity to the neoantigen peptide pools?
No, not really. It is tough to say though because we wouldn't analyze these cells that wouldn't hit a specific
Mm.
cutoff, of 50, outgrowth of 15 million cells. So um, those we have no idea if there's highly reactive TIL in that 15 below 15 million fragment or not. It very well could be. So we didn't really see anything directly. It certainly wasn't the case that the very, very high outgrowers were strongly more reactive.
Perfect. Another question that we have is, what is your working hypothesis for the cause of the observed waves in TCR dynamics during TIL expansion? Do you think this is driven by exhaustion or by another mechanism?
So I guess what I would like to be right, I can't really, I don't have a good explanation for it. So if I were to speculate, though, I do think it's a combination of exhaustion and potential epitope spreading. So what we're doing is causing some level of immunity and cytolysis in the tumor, which is then causing a bit of kind of a boosting an immune response. And so I don't know if we're getting necessarily novel TCRs out of it. The cancer has been there for years, so I would imagine that all of the TCRs that are going to react, the patient has already seen those. But we may be re-exposing candidate or tumor reactive TIL, or not TIL, but tumor reactive T cells that for whatever reason weren't reactive the first time they were exposed or weren't significantly enough reactive.
Perfect. And do you believe the CISH knockout helped in TIL outgrowth?
Yes, we do see some better expansion of CISH TIL that are CISH knockout. That's for sure. We also do see some better survival post outgrowth. It's not a massive difference, but it does improve both the expansion and if we culture them out, they do tend to stick around a little longer.
Perfect. And what kind of assays you have done or any data you have to address safety concerns for CRISPR editing? For example, off-target editing, translocations, things of the sort.
Sure, yeah. We completed kind of two different approaches for looking at off-target effects. One was GuideSeq, and one was looking at just the top 40, I believe we looked at off-target effects or off-target sites, and then used NGS to determine if we're seeing any off-target effects. Notably, we really didn't see anything at all above our limit of detection with those assays. With this particular guide, we found that we've done this with other guides as well. We find that certain guides are far worse than others when it comes to off-target effects and things like that. For translocations, we looked at chromosomal rearrangements, things like that. And again, it was a lot of them. These patients, they're generally very heavily pre-treated, and that often includes radiation
Mm.
therapy. So we would see a lot of chromosomal damage in a lot of the TIL. That said, we didn't see any increase of it with our post-stitch editing or anything of that effect.
Perfect. Have a few more questions. Since you did both the NEX and TCR repertoire, how were the two approaches complementary in this case?
Well, the one figure I showed, I think, really highlights that we were able to use our RNA-seq with the rep-seq to identify um, interesting TIL in our infusion product, our kind of post hoc infusion product analysis, and then use those TCR clonotypes to track that, okay, no, this cell right here is actually a TIL from the infusion product, and track those TCRs out 1 year in the patient, and then use the CISH RNA-seq then to go and determine that those. And it's fun to say that to speculate that they would be our TIL infusion product because of that CISH knockout. Obviously, that's not definitively shown because we can't actually look at the DNA of that specific cell, but it is consistent with what we would see if they are in fact infusion product TIL that are still outgrowing in the patient.
Perfect. And have you performed any antigen prediction on the TCRs?
No, we really haven't on the TCRs, and that's a good idea that we should do. We did do a lot of predicted antigen binding or peptide binding
Hmm.
to the MHC when we originally were looking at our neoantigen peptides and actually organized pools in such a way that we had the high CD or high MHC class 1 binders in one pool, high MHC class 2 binders in a different pool. So we could try to see if the antigens with a higher predicted affinity would have any pattern. Um, and we didn't really see any obvious pattern with that. But now that we have identified the TCRs that we think are reactive, it's definitely something we should go back and do and determine. And then, I mean, we would have to clone out the TCRs and run in kind of peptide pulse neoantigen or peptide pulse with the candidate neoantigens and show that that TCR does in fact react. With that particular peptide,
Hmm.
just to kind of validate that prediction is correct, which would be quite a bit of legwork, but it is a good idea.
Perfect. So again, thank you very much, Matthew, for such a great presentation and showing that amazing amount of work. Just a reminder that this webinar was recorded, so stay tuned for the recording to share with your colleagues and your peers. And also stay tuned for more webinars for my repertoire. And we have a lot of great topics, such as the one Matthew just presented for this. So thank you again really much, Matthew, and our repertoire team for organizing this webinar. We thank you all for attending today. Goodbye and have a great day.